and introduce yourself, that I think it'll be great for time-wise. Give you plenty of time.
Yes. Okay, let's start. Um, so my name is Haiming Kerr. Currently I'm an acting assistant professor under geriatric and gerontology medicine. So my background is PhD in rehab science, and I started as a postdoc with Dr. Jose Garcia's group. And right now I transitioned to a junior faculty. And my research has been focused on cancer cachexia and the sarcopenia, mostly related to muscle biology and the physiology, and trying to find pharmacological treatment for these conditions. And today, my talk will be focusing on the approaches to sarcopenia and targeting muscle dysfunction in aging. Okay. So a little background about sarcopenia, which is a progressive loss of muscle mass and strength in mostly elderly people. Um, and you could start at an earlier age, and it affects 25 to 45% of older adults in the United States. Um, and it has adverse outcomes, including falls, frailty, disability, poor quality of life, and mortality. And on the right is a chart showing the decline in muscle strength and muscle mass. They are not parallel to each other. And this is a study using the knee extension strength and also evaluated muscle mass using DEXA. As you can see, with aging, both lines go steeper, but the drop in muscle strength is much more prominent than muscle mass. So far, there is no FDA-approved pharmacological treatment for sarcopenia. With exercise is the only recognized intervention. And other treatment like nutrition, such as amino acid, calcium, and vitamin D, as well as pharmacological interventions can only targeting muscle mass, but less reported in improvement in muscle strength. Therefore, an effective treatment targeting muscle strength is still missing. So as I talk about, there is an unparalleled relationship between muscle mass and muscle strength loss with aging. The clinical definition of sarcopenia has been changed over the years. So particularly the most up-to-date definition of sarcopenia put lower muscle strength forward before muscle quality and quantity, and followed by the decrease in muscle mass or quality. And lastly is using lower physical performance as the third definition. So on the right, this is the common diagnosis clinically and the cutoff points for men and women. And these are the common tests in humans. So um, what are the molecular and the cellular mechanisms contribute to sarcopenia? So, for people in aging field would know this framework of hallmarks of aging, which is composed of 12 different processes contribute to age-related dysfunction across different tissues. And this is a very nice hallmarks for all kinds of aging study, including sarcopenia. And recently, there is a review paper utilizing this hallmark map and also use the previously mentioned diagnosis of sarcopenia in human, and they find all these hallmarks has changed with sarcopenia. And among all this aging hallmark, mitochondrial dysfunction appear to be the most closely associated with sarcopenia. And this is associated with like decrease in OXPHOS genes and decrease in electron transport chain and changes in TCA cycles and also decrease antioxidant and increase the oxidative stress. And meanwhile, the other hallmark process also direct or indirectly connected, such as chronic inflammation or genetic changes, and also loss of proteostasis or macrophage changes may contribute to a loss of mitochondria, and also nutrient sensing also contribute to the metabolism changes in mitochondria. So overall, this uh, shows us mitochondrial dysfunction is central in the progress of sarcopenia. So to further understand how to counteract the sarcopenia and using an animal model is very important for preclinical study. Because mice model would allow genetic or drug intervention, and also mice age is very similar to human, but much shorter time for preclinical studies. And so far, the natural aging model is still the most optimal model for sarcopenia study. However, most studies only select one older age group and confirm that is sarcopenia only using the loss of muscle mass, but not muscle function. And up to this point, there's no standard assessment for sarcopenia in preclinical study, like in the human studies. So therefore, my first study is to utilize natural aging mouse model to develop a sarcopenia model, and also utilize the equivalent human age from 70 to 80, which is 24 to 30-month-old mice, to establish a preclinical sarcopenia definition. So there are three folds of the study, and the first is to define sarcopenia in C57 black 6 mice using clinically relevant criteria. And second is to understand the relationship between the functional loss and the muscle mass during the progression of sarcopenia. And lastly, to elucidate selected molecular mechanisms underlying this progression of sarcopenia. So very similar to the clinical sarcopenia definition, we also tested three components in mice, including forelimb grip test, which mimicking the hand grip strength. And we also evaluated high limb muscle mass, which is equivalent to the muscle quantity and the quality, and treadmill running time, which is putting the mouse on the treadmill and record the time to exhaustion. And this is mimicking the physical performance. So we evaluated both young mice and aged mice, and we used the young cohort as a standard. So we have the mean of the young and use 2 standard deviation below the mean as a cutoff. So below the cutoff, the mice were considered as having either low muscle strength, low muscle mass, or lower physical performance. And if any of these mice showing one deficit among these criteria, we consider it as a probable sarcopenia. And if it shows 2 to 3 deficits among these criteria, we consider it is a sarcopenia mice. And versus no deficit is non-sarcopenia. And with this definition, we evaluated male C57 black 6 mice, aged range from 23 months to 30 months, as I mentioned, equivalent to 70 to 80-year-old humans. And as you can see, the prevalence of sarcopenia increase with aging in mice, and with equivalent age of human, it shows 90 to 22% of sarcopenia incidence in 23 to 26-month-old mice, which is very similar to 10 to 27% in equivalent age of humans. And to further estimate sarcopenia, in these mice, we want to understand the relationship between muscle mass and muscle function. And here are the more accurate measurements of uh, muscle mass and muscle function using the cross-sectional area of the individual fiber, myosin heavy chain fiber types. So in skeletal muscles, it has 2A, 2B, and 2X fibers in mice, and 2B fibers is the fastest twitch fibers among all three. And we do see a decrease mainly in 2B fibers, and there's a trend of difference in the sarcopenia mice compared to the young mice. But these changes is not in 2A or 2X fibers. And when we look at the muscle force generation, which reflecting muscle strength, and we use different force stimulate muscle and have a force generation curve. And we do see a decrease in probable sarcopenic mice and also sarcopenia mice. And this indicating that the decrease in muscle strength actually happening earlier than decrease in actual cross-sectional fiber area. So as we talked earlier, the main mechanisms contribute to sarcopenia is mitochondrial dysfunction. So therefore, we looked at mitochondrial respiration and mainly stimulated by complex 1 dependent respiration. So we look at the ATP stimulated maximum respiration and also FCCP stimulated, which is uncoupled maximum respiration. And interestingly, we do see age-related decrease in both mitochondrial respiration, but there's no significant difference across the sarcopenic groups. And we further evaluated the complex 1 to 5, which are the OXPHOS complex components, and we see they have a very similar change, and they only decreased with age, but there is no difference across different sarcopenic groups. And to further evaluate other mitochondrial components, we look at mitochondrial biogenesis markers, PGC1 alpha. And interestingly, we do see a gradual decrease in PGC1 alpha level with the progression of sarcopenia. And also, we look at the overall protein level in isolated mitochondria, and it mainly decreased in sarcopenic mice. And similarly, SDH staining also reflects the mitochondrial content, particularly in complex 2 related oxidative capacity, and this is lower in probable and sarcopenic mice. Another process very important to maintain mitochondrial quality is called mitophagy, which is autophagy process by tagging the mitochondria with ubiquitin through adapter proteins such as P62, and then the lysosome can engulf mitochondria and degrade the damaged mitochondria. So this is considered as a quality control process for mitochondria. So we examined a few markers in this process, including the P62 and LC3, which is the marker of the lysosome. And the gene levels is shown here. And we see a gradual decrease in these two levels, and the main difference is between the young and the sarcopenic mice. And at the protein level, we have seeing a very similar decrease, mainly in the probable sarcopenic and the sarcopenia mice. And lastly, we will look at the AMPK, which is the upstream regulator for this process. And very similarly, the decrease is mainly in the sarcopenic mice. So before, all I talked about was in the male mice, and what about in the female mice? So for the female mice, we examined a smaller cohort, which is aged from 27 and 28 months. So we use very similar criteria for female mice. So we use the female young mice as the standard and use two standard deviation below the mean as a cutoff. As you can see, it's very interesting. All three parameters decrease at the same extent, which is different than the male mice. If I can remind you here, that the decrease in grip strength and muscle mass is much less than in treadmill running time, but they are all about the same in female mice and much less than in the male mice. And when we look at the sarcopenic status, also in female mice, it shows less incidence of sarcopenia mice compared to the same aged males. And similarly, when we look at the protein levels of the mitochondrial biogenesis and the mitochondrial proteins, as well as the field markers of mitophagy, we do not see the similar changes with aging or with the progression of sarcopenia, which indicating a different process in female mice. So in summary, this part of the study we found muscle mass and function decline at different trajectories while male mice aging, which is very similarly to what we found in the clinical studies, with muscle strength decrease more severe than the muscle size. And male mice also demonstrated several mitochondria-specific changes, including depressed oxidative capacity, AMPK-driven mitophagy, and PGC1 alpha-mediated mitochondrial biogenesis. Although these processes occurred at different times of the progress of sarcopenia, and it happens earlier for the decrease in mitochondrial respiration and oxphos content, even without the symptoms of sarcopenia. And lastly, we found female mice has a much milder sarcopenia phenotype compared to the male mice, and with minimum change with these selected molecular and cellular changes, which may partially explain the sex-specific difference. So our next question is, what would be a good treatment for counteracting sarcopenia and targeting mitochondrial functions? So as our group has a long-term interest in ghrelin and the ghrelin receptor signaling, so we look at the ghrelin signaling pathway with aging and sarcopenia for our next study. So a little bit background about ghrelin and ghrelin's receptor. Ghrelin is known as a hunger hormone secreted by stomach, and it binds to GHSR1A, which is also the only known receptor for isolated ghrelin. And GHSR1A is enriched in pituitary and the brain, and it's not expressed in skeletal muscles. And the ghrelin is able to bind to GHSR1A and activate it to release growth hormone and also increase appetite and further contribute to adiposity and increased body weight. So far we know with aging there is an increased circulating ghrelin and GHSR1A. And also, previous studies has shown ghrelin deletion is able to improve endurance and grip strength in aged mice. And this is also associated with AMPK, PDK4 signaling, which is also a signaling closely related to mitochondrial function and metabolism. So the ghrelin receptor GHSR knockout mice is also established in the aging field, but mostly known for its effect on decrease in age-related adiposity, reduced energy expenditure, inflammation, and insulin resistance, and its effect on skeletal muscles is still less known. However, the ghrelin receptor knockout might be a more viable target for sarcopenia than ghrelin deletion because it's a receptor and it's a more druggable target. So therefore, the second part of the study is to determine whether genetic deletion and pharmacological inhibition of GHSR1A can mitigate sarcopenia, especially targeting the functional loss in skeletal muscles and explore the underlying mechanisms. So very similarly, in this study, we utilized the wild type and GHSR1A knockout mice with a C57 black 6 background. And we started with male mice as we've seen all these molecular changes in male mice and also the established rationale are mainly based on male mice. And we evaluated the sarcopenia measurements, as I mentioned before, including grip strength, muscle mass, and treadmill running time. And we utilized the previously established cutoff for this cohort of mice. And lastly, we analyzed the molecular markers in skeletal muscles. So first of all, we look at these sarcopenia measurements in these mice. And with aging, as you can see, there's a decrease across different groups. But this decrease is attenuated in 24-month-old knockout mice in terms of grip strength. And for treadmill running time, it also improved in the 24-month and the 28-month knockout mice. And for highly muscle mass, knockout mice showing a lower muscle mass when they are younger, but we do not see a genotype difference between the wild type and the knockout while they're aging. And then we use the similar sarcopenia criteria to identify sarcopenic status in these mice. And we do see an increased number of non-sarcopenic mice in the knockout mice. And this number is reduced in the most severe sarcopenia mice. Although there is some differences in the probable sarcopenia mice, which is less in knockout mice, but more in sarcopenia mice with the knockout. And similarly, we further look at the muscle contractility using the force frequency curve. And we do see a decrease with aging in both genotypes, and there's no genotype difference. And the peak force generation is the peak level of these curves. And we only see an age-related decrease, but no genotype difference. And the specific peak force is normalized to the physiological cross-sectional area, which reflecting the muscle quality. But we do not see a difference across different groups. And also the decrease in these muscles with aging are equivalent between genotypes. But anyhow, when we look at the fatigue test, which is using electric stimulation and stimulating skeletal muscles every 2 seconds, and the force generation is decreased over the time, and we do see a genotype difference in the absolute force generation and also in the normalized force generation. And this is happening both the old mice and also in young mice. So this indicating that at least the fatigue resistance is altered by GHSR1A deletion, and this is my partially explain their difference in endurance. So similarly, we examined mitochondrial respiration using the seahorse technique, and we do see a decrease with aging in both genotypes, and then there's no genotype difference, and the OXPHOS components mainly decreased in wild type, although knockout has no difference than the wild type. But interestingly, we do see a decrease in the citrus synthesis activity, which is reflecting the total mitochondrial contents with aging in wild type. But this is not in knockout, and in knockout, it has been improved in the aged mice. And lastly, the SDH staining here reflecting the decrease is more dramatic in wild type mice, although there's no genotype difference here. And the other mitochondrial markers involved in mitochondrial biogenesis, we look at the PGC1 alpha, and the decrease in wild type is we see age-related decrease, but we do not see the difference here in the knockout mice, and also in the aged knockout mice, we see improvement in PGC1 alpha. And similarly, we see increase in SIRT1, which is upstream regulator of PGC1 alpha. And also, we do see a more dramatic decrease in mitochondrial DNA, and we do not see the similar changes in knockout. So this indicates that GHS1 deletion has an effect on mitochondrial biogenesis and mostly benefit the progress of making mitochondrial DNA. And lastly, as we're also interested in the mitophagy progress, so mitophagy, as I mentioned before, is a mitochondrial quality control. So when there is a damaged mitochondria, and there will be a change in the mitochondrial membrane, and the pink one is able to activate by these changes in mitochondrial membrane potential and bringing PARK2. Parking is E3 ligase, which is tag the ubiquitin to the mitochondria and further regulated by the adapter P62 and bring it to the lysosome and into the autophagy process. So in that case, we evaluated these markers in the isolated mitochondria and the total protein level. And interestingly, we do see an increase in the P62 level in the 24-month-old knockout. And also, we do see an increase in pink1 level in the OH, the knockout mice. And when we look at the parking and to the VDAC, which is the mitochondrial membrane protein, we also see an improvement in the knockout mice. And lastly, the colocalization of TOM20 and LC3, which is also a mitochondrial membrane marker and autophagy marker, is the colocalization is improved in knockout mice. So in summary, we do see these elevations is in this process of mitophagy and mostly in the aged knockout mice. So the next step, we want to look at a little further, a bigger picture of this molecular changes in the GHSR1A deletion regulated sarcopenia. In that case, we use the skeletal muscles from both wild type and the knockout mice in old and young mice, and we did proteomic analysis. Um, so we evaluated the proteins and we did a correlation between the individual protein and the sarcopenia measurements. However, we did not see any correlation at the individual protein level. So in that case, we move forward to pathway analysis. So here we use the commonly used the CAG pathway analysis. Then we perform the correlation between the pathway and the sarcopenia measurements, either the treadmill running time, grip strength, or the muscle mass. And we did interaction term of the age and the genotype, and this showing the perturbed pathways in each of these correlated sarcopenia measurements. So interestingly, we only see one pathway is overlapped across the three measurements, which is the complement and coagulation cascades. And this pathway is likely related to immune response and also like muscle fiber regeneration, and thus contributes to all components of the sarcopenia component. And the overlaps between the grip strength and the muscle mass here are mostly related to the protein homeostasis and amino acid homeostasis, which is as expected because they contribute to muscle mass and further muscle strength. And the overlaps between treadmill and muscles, although it's called a diabetic cardiomyopathy and thermogenesis, but mostly still related to mitochondrial function proteins. So here we also mapped out the top 5 proteins of each pathways. As you can see, most of them are related to mitochondrial function, and secondly, they're related to extracellular components. So to further identify where are these top 5 ranked proteins, we use the gene ontology cellular component term, and we do see the most of the proteins still located at the mitochondria in this process, and followed by the cytoplasmic proteins and extracellular proteins. And then we further took the mitochondrial protein from these top 5 ranked proteins and then did another functional analysis using gene ontology to identify biological function. And we identified that these proteins are closely related to mitochondrial respiration and energy metabolism, and the mitochondrial substrate transport and membrane function, oxidative stress, fatty acid and amino acid metabolism, and lastly, mitochondrial biogenesis, which is partially in line with our findings in the protein levels. I see there's a question. Should
Oh, no, I mean, you're using the term proteomic analysis, but these are gene pathways. This is
Yes,
transcript. Yeah, I would, I think that term should be reserved for proteomics, I think, not inferential proteins from genes. Correct me if I'm wrong.
yes, you are correct.
Especially when we're talking about extracellular matrix and things like that. There really is it's really suspect as to how much correlation you're actually going to get at the protein level.
Yeah, it is true, but the problem is with our proteomic analysis, the tools for pathway analysis is very limited. That's why we try to like utilize the different tools to analyze it. But I totally understand for the proteomic analysis, we have to take into account of the post-transcriptional effect. Um, yeah, but thanks for the advice. And lastly, we look at these lifespans of the wild type versus the GHSR1A deletion mice. And in terms of lifespan, we do not see a significant difference in their survival rates, which indicating that although GHSR1A deletion benefits muscle function, but it didn't affect lifespan. And in addition to the genetic modification in the GHS1A, we also look at pharmacological inhibition of this target. So PF 5190457 is a GHSR1A inverse agonist, first developed by Pfizer, and it inhibits constitutive activity of a GHSR1A and completely block its activation by acylghrelin. So this drug is under phase 1B clinical development, but mainly due to its effect on reducing alcohol craving. So this is the article about this clinical investigation, and the individuals were tested in this bar-like lab environment. But in our study, we are interested in its effect on inhibiting GHS1A, and we found that based on literature in male rodents, it attenuates either ghrelin or ghrelin mimetic hexarelin-induced food intake. And we utilize a dose which is well tolerated in rats and has minimal effects on locomotor activities. So with this drug, we tested 9 to 11-month-old C57 Black 6J male mice, which is about the middle age as the mouse age. And we treated with either the PF drug versus DMSO for 28 days. And we measured weekly function and also harvest the skeletal muscles to evaluate muscle mass. So very similar to other previous investigation, we do see a decrease of cumulative food intake over the time in these mice. And also mice treated with the Pfizer drug has lower body weight and reduced fat mass compared to the DMSO control group, while they have increased treadmill running time at the end of the study. And even with treadmill normalized with body weight, we still see this difference. However, it didn't change the grip strength over the time and also did not have an effect on muscle mass. So at least it indicates that inhibiting GHSR1A pharmacologically partially mimicking its effect on its genetic models. And lastly, we evaluated several similar markers, including PGC1 alpha and the SIRT1 nerve1, and these are the transcriptional factors of PGC1 alpha. And we do see increase in the PF treated mice in PGC1 alpha. And similarly, we tested the mitophagy markers, and we do see an increase in LC3-2 and BNIP3, which is another mitophagy pathway independent of the pink PARK2 pathway. So in summary, we do see a very similar changes using pharmacological intervention targeting GHSR1A as what we've seen in the genetic model. So in conclusion, our study used the deletion of GHSR1A model, and we do see an improvement of the muscle function and the severity of the sarcopenia without affecting the lifespan in C57 black 6 male mice. And we do see a similar effect from the pharmacological treatment targeting GHSR1A, and also it has an effect on food intake and reduced adiposity, although its effect is mainly on endurance rather than grip strength. And for the molecular pathways, both interventions has shown a modification in mitochondrial biogenesis and mitophagy. But actually, the causality remain to be confirmed in this process. And lastly, the study provides a very strong rationale for future research targeting GHS1A inactively as a therapeutic strategy for sarcopenia. And for future studies, we should also incorporate female mice, as we have seen sex differences previously in the wild-type mice, and they have different progression of sarcopenia and molecular process. So here is acknowledgement and thank you for like everyone in Garcia Lab for this study, and my mentor Jose Garcia, and also people from Nathan Schuck Center on helping the proteomic analysis, and people from the VA course. So that is for my presentation. I still have plenty of time for questions.
Can you see the questions, Haiming? Because there's one from Kay, and it's
Yes.
okay.
So she asked, is there any connection between effects of ghrelin on muscle quality and glucagon-like peptide 1 effects it might have on muscle development or loss? So I'm not very familiar with the effect of glucagon-like peptide 1 effect on skeletal muscles. But I do believe the GLP-1 drugs can affect muscles, like while you're losing body weight and also lose muscles. But I never really compared it to the effect of ghrelin. But somehow I think there might be some similarities, as ghrelin is able to cross blood-brain barrier and has some orexigenic effect. So reversely, for our model of GHS1A, it could affect the whole process centrally. As we've seen the pharmacological process, there is a decrease in food intake and body weight. So I do believe there is some similarity between the two. I hope I answer your question.
I mean, great talk. I had a question, and I'm sorry if I missed it in your background or don't remember from previous talks, but so ghrelin is a hunger hormone, correct? Is that
Yes.
what? Yes. And so what is its specific action within the muscle? Is it to trigger the muscle to take up energy or?
Yeah, so the effect of ghrelin on muscle is kind of complicated, and as I mentioned before, its receptor is not expressed on skeletal muscle. So but that's the only receptor we know. But there might be an unknown receptor of ghrelin. It could be in skeletal muscles, because there is also like effect from like unisolated ghrelin, and our previous model using GHSR knockout mice, we still see an effect of ghrelin on skeletal muscles. But the known effect from ghrelin on muscle adaptation mostly from like energy expenditure,
Mm.
as we know, and its effect also on inflammation. So that's why we see a beneficial effect of ghrelin most in like severe muscle atrophy, like a cancer cachexia. So which is kind contradictory to this talk, like in the process of sarcopenia, which is a more chronic muscle atrophy process.
Thanks.
Um,
And then I
yeah.
also had just a technical question. I noticed in your last study you used aged mice that were either 24 months or 28 months. And I'm wondering if that's because that's often when they kind of convert to the sarcopenic state. And so you can catch mice in the non-sarcopenic stage, but still aged at 24 months. But by 28 months, they're more prevalent in the sarcopenic stage. Is that correct?
Yes. Yeah.
And what's the typical mortality at 28 months? So how do your mice typically survive 100% up to that 28-month stage, or do you have to buffer in for attrition?
Yeah. So I believe for C57 black 6 mice at 24 months, the survival rate already dropped to 50%. Um, and it might be variable with different like your resource of the mice, but it's very similar. So we do lose a lot of mice at 28 months. So unfortunately, that's a good point. Like our study is cross-sectional study, so it may have some like survival effects when we detect these molecular changes. So whoever maybe already have these molecular changes happen earlier probably already died at that point.
Yeah, that's a good point. Thanks.
I have a question about the sex steroids in mice as they get older. So do the males, I guess the females mimic what happens in humans, right? After a certain age in terms
Um,
of sex steroids?
in mice,
Yeah. Do we know?
um, I think mice might be different than humans.
Well, yeah, I'm just wondering about the trajectory of loss of the sex steroids
Uh,
in
as
female versus male C57 black 6. I know
right,
that, yeah.
because I believe mice they I'm sorry, I'm not very familiar with that topic though. Um, but I think mice after a certain age, then they dropped dramatically in the sex steroid, but they still exist. Um, which is I think much earlier than human age. I didn't look at the actual um equivalent age for that though.
No, I'm just thinking in terms of if you see sex differences in muscle or sarcopenia, I imagine it's going to
Right.
come up. And I'm just Googling it real quick, and there is a body of literature. I just wasn't familiar with it. So I think it'll be something else to consider.
Yeah, I do believe that contributes hugely to the sex difference. Even when the female mice, they're already and they're like their fertile level after a certain age. Um, but they still the sex hormone still plays a huge role even at the later age.
Right. Yeah, that's because I remember we
Yeah,
used to always have to separate our
yeah.
male mice, even the older ones, from fighting, which we didn't have to do with the female mice. I'm just wondering, in terms of influencing your pathways and your sarcopenia, what those influences might be. Just wondering.
Yeah, I think another component I didn't talk about in this talk, sarcopenia, but in human is very common to have like a sarcopenic obesity. Um, so in our male mice, the obesity is um disappear when they are age after a certain level. So they have increased body fat at a certain level, then they decrease dramatically. So at the point we look at these mice, they already don't have the obesity effect. But in female mice, they still gaining weight at the same age. I think that may partially contribute to the sex difference as well, but also that's closely related to the sex hormone. I hope that partially explained.
Yeah, yeah, very
Yeah.
complicated system. No, thank you.
Yeah.
That's excellent work you're doing. Um,
Yeah, I should have if I have more time, I should have shown that that's very interesting, probably my next talk. Sarcopenic obesity.
yeah, that's always a slide now on frailty. Any frailty section is going to have a slide on that in humans as well. And
Yes,
now we're all
yeah.
confused because so many of the people who had sarcopenic obesity are now taking GLP-1 receptor agonists. And as Kate pointed out, yeah, definitely worse a loss of muscle relative to fat in humans. And yeah, it's all getting very complicated.
Yeah, I know it's also like a huge development, like counteracting GLP-1 drugs right now. So it's like just taking more pills.
Oh, and then it's now pills. Yeah, making it much harder to track who's doing what.
Yes.
My least favorite, yes, Kay saying my least favorite drug. Yes, agreed. Agreed. Um, any other questions? Really great work.